So I'm going to be talking today about people in here, about the vast diversity of phage genes of unknown function that we're discovering constantly. Let me draw your attention, if this works, okay, to two phage proteins. The first is OCR. It's a DNA mimic. It's a protein that actually mimics the fold of DNA. It binds to restriction enzymes and inactivates them. The second is this anti-CRISPR. Same thing. It's a protein that mimics DNA, binds to Cas9 and inactivates it. Now, in both cases, you have a phage-encoded effector that blocks some sort of host defense inside the cell and causes the virus to be able to kill the cell. But I mean, of course, restriction enzymes were the foundation of the recombinant DNA revolution of the 70s. And we're pretty much living through the CRISPR revolution right now. So the question becomes, if we could systematically find these phage-encoded proteins that interact with host mechanisms, could we pull on them to discover new biology? The problem with this is it's very hard to figure out where to start. The phage pangenome is vast. As we sequence more and more genomes, what you can do is you can line up different genomes of the same species, define core genes as these genes that are always present, and then define these genes that are sometimes present and not present others as accessory genes. As you add more genomes to your set, the number of core genes will stabilize by definition. But then the pangenome, which is the total genetic diversity associated with the host, that just keeps growing. So that's not saturating, right? So there are thousands and thousands of phage genes available for study, and we don't know where to start. So which phage proteins do we test first? I want to show this picture from T-even bacteriophages for a second. This is a very old observation. These genes, these IP genes here, they were amongst some of the first phage-encoded effectors known to target host processes. And the details aren't important, but these are various different phage genomes lined up. You see that while this IP gene content is being swapped in and out between the different genomes, the flanking genes are always the same at this locus. This will be better. So what this is essentially an evolutionary hotspot in the phage family, right? And our approach is based on this idea that phage accessory genes, which are not essential, sometimes present in some genomes, sometimes not in others, in an evolutionary hotspot, subject to more evolutionary pressure, they might be more likely to interface with host biology than core genes. Now, I won't be able to go into this in detail at all, but I wrote an algorithm to identify exhaustively all possible accessory genes from a phage genomic database, crucially without clustering the genomes a priori, which is hard and always leads to problems in this system. And the key thing that sets this method apart is that if you do this, now you're not just limited to studying homologs of something that we know something about or neighbors of something that we know something about. We're simply leveraging the evolutionary characteristics of these genes and asking, what else is out there? Once we have a list of candidates to test, this is essentially the experiment we want to do. You have, let's say an anti-immune gene, which blocks the activity of some sort of phage exclusion system in a host, which would otherwise have prevented a phage from infecting. So you essentially express a whole host of these genes one at a time in a variety of wild bacterial strains and ask, can the phage still infect? So here's a screen that we built to do this. We conjugate a barcoded library of phage genes into many wild bacterial strains. We do this in triplicate, and then we challenge each mixture with each phage one by one. We make 3 measurements for each infection. One at the start to figure out where things were just at the beginning. One with just the phage gene expressed, no phage added, to see what happens when you just express the SORF. And then the third with the phage gene added and a phage added as well. We did this with a pilot library of 200 phage genes that were selected from a larger set of 1,800 viral genomes of enterobacteria. Again, to test the method, we picked 20 wild E. coli strains from the ECO reference collection that was assembled in the 80s. And we challenged these mixtures with 8 model phages that we got from VEGA. Now, I won't really be able to go into the details of anything at all in the interest of time. These are all the kinds of things that we're finding. The key here is to think of this library of phage orfs as a drug discovery library. Really think of it that way. What you get will depend on the stress that you use in the screen. So because we challenged these libraries with phages, what we tend to find are anti-phage defense mechanisms. I'm going to quickly take you through some of the things that we're finding, and then maybe have time to talk about the kinds of things I want to do next. First, we find phage-encoded ORFs that are targeting conserved host pathways that we don't usually think about when we think about phage defense, at least in the circles that I operate in. So this is our most dominant class of hits, right? We have at least 3 phage ORFs that are removing some sort of chemical decoration on the O antigen, which is this long polysaccharide layer that sort of sticks out off of the LPS of the outer membrane. And that changes very drastically what phages can and can't infect. We're trying to figure out what the exact nature of this conversion is, but we think this is a new mode of phage-encoded sort of seroconversion of the host, because everything that we know of so far uses very clearly enzymes that have O antigen modifying domains and have usually been co-opted from the host and things of that nature. Yeah, so we think this is a novel mode of seroconversion of bacterial strains. The importance of these barrier defenses is further underscored in the second class of hits that we find. So this is what you canonically think of it as phage-encoded abrogation of host immunity. It's a phage protein that disables the defense locus. The key thing is that here we couldn't actually see this by our transposon screening efforts until we peeled off the outer layer of immunity. Right? So we have to remove the O antigen first, and then suddenly there was a second layer that was revealed, where, for instance, this new ORF that we have, ORF 143, disables this defense locus. This actually turns out to be the answer to a decades-old story of T-even phage host coevolution. Again, I won't really have time to go into the details, but this is the immune system that ORF 143 that I described disables. It was previously known that phage T4 could bypass this immune system through some unknown mechanism, and then that's this ORF. It's a T4 encoded protein. And if you thought that these restriction modification systems were boring, we're finding new activities of RM systems that would have been missed if you hadn't done this sort of phage forward approach, right? So what I mean by that is here are known inhibitors of restriction modification that seem to be being detected by these RM-like systems that then trigger cell death. So this establishes this, there's this model in the field of guardian immunity, right? The idea being that you have a phage, it encodes an anti-immune effector, it blocks some sort of frontline defense like an RM system, which would otherwise have stopped the phage. But then there's this other layer of immunity that detects the presence of this phage factor, this offending phage factor, and kills the cell, saying the infection has gone too far, we need to protect the colony. But when you think of RM systems that are laying traps for their own inhibitors, you have a multifunctional immune system that can respond dynamically as the infection proceeds. If the phage DNA enters the cell, try and kill it. If it bypasses that effect and starts making these dangerous proteins, trigger a programmed cell death because the infection has gone too far. So the model that we had here kind of evolves another step, and what you get is this coupled targeted plus abortive immune system. And this, like many things in the field, is something that Kira and Eugene had hypothesized, I think, 10 years ago, and we're very excited to be seeing examples of this in the field. Another RM-like system that we found, this one's really interesting. It also has that ability where it detects the presence of its own inhibitors and kills the cell. But if you look at the actual organization of this locus, it doesn't actually have the endonuclease. So this is a restriction system that should not be able to do what restriction systems do, which is detect foreign DNA and kill the cell. And that evokes this idea of decoy immunity, which is very widespread in plants. So our model evolves another further step where now this coupled immune system has lost the ability to target the phage at all, and now only exists to detect the presence of the inhibitors of its ancestors. So, that was quick. Again, the details aren't super important, but what I'm trying to sort of showcase here is this platform lets you paint a somewhat holistic picture of anti-phage immunity in any given niche. We've seen barrier defenses and inhibitors thereof. We've seen traditional immune systems. We've seen guardian immunity, which is immunity triggered by the presence of immune inhibitors. We've seen this guardian immunity combined with targeted host defenses. We have viral proteins deployed in antiviral defense. I couldn't talk about that. We have decoy immunity, and then interphage competition, which again is here, and I'm not going to go into it. Suffice it to say that we found instances of this. Now I've been trying to figure out kind of where to go from here. What phage library to clone next, where to sort of direct this platform, what host to move towards. And the thing that I want to emphasize is that even though we did this with 200 genes, right, we can easily handle 20,000. It's no different from doing a transpose on screen. The number of elements is far lower anyway. So if you want to systematically investigate anti-phage immunity in a host, I would propose that what you'd want to do is cast a wider net, right? So the key thing here is, and I think this presents a really good opportunity, E. coli, Salmonella, and Klebsiella are very different beasts, and we have hundreds of clinical strains. But their phages, if you start playing with their genomes, they tend to cluster together. So what you could do is you could build one phage ORF library. You could do this with only accessory genes, or you could take everything, which wouldn't be that much. It would be about 20,000 factors. And you could test the same library in multiple different strains of pathogenic bacteria of interest. And perhaps then you start to see either convergences, different phage factors that have the same effect in multiple different hosts, or divergences. What are the kinds of things that we could look for? I mean, of course, we'll find instances of phage factors disabling host immunity. And thanks. And I'd argue that this is what you would really want to see, because if the virus cares about it, then you know that you're targeting a bona fide immune system. We'll find triggers of abortive immunity. We've talked about these extensively. And you would also find triggers of abortive systems. We didn't see these in the screen, but it's a protein detection problem. Let's say you express a phage capsid inside a host. Is there something in the host that understands what phage capsids look like, or understands what phage polymerases look like? How diverse is the set of these protein detection systems that exist in bacteria, right? Like, is there an endless diversity? Is there a programmable version of these? And then finally, and this is, I think, what I'm most interested in, if you look at different host phage niches, right? The popular ways of keeping phages out are things like CRISPR systems, right? These are conserved, we know what they are. How long is the tail? If you look in E. coli, do you tend to find phage exclusion systems that only exist in E. coli and not in Salmonella and not in Klebsiella? How much do you have to probe before you can start to get towards something that Vivek mentioned in his talk as well? Being able to predictively talk about sort of a phage host interaction purely from the genome. And then finally, just sort of coming back to this idea that it's not just phage defense, right? Once you have a phage ORF library, these viruses have evolved over 1 billion years to mess with host cells. You could, in principle, use any stress that you were interested in finding, host sort of vulnerabilities around. So, really think of this as a drug discovery screen more than anything else. And we've done this with anti-phage immunity here, but we hope to expand this significantly in the future. Thank you for your time.